This Week in Evolution, episode number 1, recorded on September 29th, 2015. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiEVO, the podcast all about the biology behind what makes us tick. Joining me is my co-host from Salt Lake City, Nels Elde.
Hello, Vincent.
Hey there. How's it going out there?
It's going really well. That theme music really gave me the chills just a second ago.
We're starting a new podcast, Nels and I. We're going to tell you all about it. And it's exciting. You know, I've done this before, but let's see. This is my 5th podcast, right?
Wow, that's impressive.
I had a few that never got going, but this is the 5th. Yet it still is very exciting. And this is something that germinated back in July of 2015 with Nels.
Yeah.
And as you know, the name is This Week in Evolution, so we're going to talk all about evolution and how to study it. You may know Nels because he's been on TWiEVO before. He was on TWiEVO in Denver, right?
Correct. That was a couple years ago at the American Society for Microbiology's annual meeting.
Yep. You were younger then.
Time flies when you're having fun.
So this is an interesting combo. I'm an old guy, and you're a young guy.
Well, that's being generous.
I think you're young. You're
you.
a lot younger than I. Some of you who follow me on social media may know that Els was the recipient of the Anne Palmenberg Junior Investigator Award at the American Society for Virology meeting this past July. And I had the pleasure of introducing you as one of 2 recipients of that prize. And you came up on stage and you took a selfie of us, right?
Yeah, that was really fun. And I think actually maybe we can trace that moment to the origins of our new podcast, TWiEVO.
You know, when you did it, I thought, this guy is okay.
That was a really fun moment. And what an honor, by the way, to receive that award from the American Society of Virology. It was really great.
Fully deserving. And that's why we're doing this podcast together. I can't think of a better person to do this with you. And so that's the first one. You know, I think we should start by having you tell us just a little bit about yourself, even though we did it in Denver. Mm-hmm. Let's do it again. Where are you from originally?
That sounds great. So I'm originally from Minneapolis. I grew up in Minnesota. And as an undergrad, I attended Carleton College, about an hour south of Minneapolis. And that's actually where I trace my first interest in evolution, and certainly genetics, was to a great professor and mentor, a fellow named Stefan Zweifel, who sort of took me under his wing and got me really fired up about genetics. We were thinking especially about model systems, so the budding yeast, Saccharomyces cerevisiae, and that kind of lit the fire for me.
And Salt Lake University... University of Utah, right?
That's correct.
In Salt Lake City, Utah. This is your first faculty position.
It is. So I opened up my lab about 4 years ago now, a little more than that. 4 years, 3 months, but who's counting?
4 years, wow.
And so between, Minnesota and Salt Lake were some stops in Chicago, where I got my PhD at the University of Chicago in cell biology and molecular genetics. And then after that, some years as a postdoctoral fellow at the Fred Hutchinson Cancer Research Center in Seattle, Washington.
Who was that? Who was the postdoc with?
So that was with a guy who you've run into a few times on TWiEVO, Harmeet Malik.
Absolutely. Were you in the lab with Sarah Sawyer?
Yeah, I was. So another TWiEVO semi-regular. Sarah was Harmeet's first postdoctoral fellow. He had just launched his lab a few years before I showed up. Sarah came right as he started his lab, and she and I overlapped for about a year and a half or 2 years.
We have to have some guests in the future on this podcast.
Most definitely.
And Harmeet should be one of them, even Sarah too, right?
I agree.
So our plan is to do this for a long time, right?
Yeah, absolutely.
If people listen.
We'll let the people decide.
And the cool thing about it is you're there 4 years, so at some point you're going to come up for tenure, right?
That's the idea. So the tenure clock here at the University of Utah is pretty similar to that in other universities. At about the 6 or 7 year mark, the tenure decision comes through. And so I thought, in addition to thinking about some of the cool work in the field of evolution, that we could also sort of think about the life of science and use this as maybe a podcast diary as I go through some of those steps.
I'm sure you'll get tenured, man.
Well, we'll find out, won't we?
And when you do you think, so right now the plan is to do this once a month, right?
Yeah, that sounds perfect.
So if you get tenured, when you get tenured, do you want to increase it a bit?
Yeah, I will make that commitment today. We can,
all right.
Once we get through that small and interesting obstacle, let's see where we go.
Yeah, from time to time, you should tell us what's going on, So.
Oh, I'd love to.
I've been through it.
Yeah, was that a few years ago now?
Yeah, the 90s, something like that. It's a long time.
Yeah.
I was going to ask you one other thing. Oh, darn, I forgot. See, this is the problem. Here it is. First of all, if you don't like Nels' sound, it'll get better next time because his mic is ordered, right?
That's right. Yeah, so as a podcasting rookie here, I'm still getting my act together. So there's a microphone on the way.
Good.
Yeah.
And we'll bring you up to the Twix speed of great sound.
I look forward to it.
So don't worry about that. Nels, why do you want to do a podcast?
Great question. I think that, there's a number of things that go into it. One is that the topic that we've chosen, evolution in particular, I think is at a really interesting place right now, where we have this sort of confluence of many different disciplines: experimental biology, broadly defined, virology, a topic of interest that you and I share, microbiology, cell biology, genetics, all kind of organizing around evolution. And so I think it's a fun time to sort of look at the playing field of what's happening and sort of lift up some of these studies and approaches to share with folks about why biology is such an active and fun thing to pursue.
Is there enough work to keep us busy?
Yeah, I think we could. I think we could go for another 4 billion years to mirror how much time life has passed on Earth in estimates so far. Plenty of stuff to do.
So you would what do you call yourself? An evolutionary biologist?
Yeah, I call myself an evolutionary geneticist.
Geneticist, okay.
And also a cell biologist. And so it's kind of these mergers, again, of these disciplines that have really fueled my interest. Another point to bring up, Vincent, given your great sort of record of podcasts, is, as you mentioned, I was on TWiEVO a while ago, the virology podcast. You also featured some of our work on TWiM this week in microbiology. And so I kind of got in my mind this idea that if I could pull off a TWiEFecta, three TWiV podcast categories, that would be a great achievement. Maybe that will push me over the top for tenure.
Are you working on some parasite problems?
Well, we are.
Good.
For TWiP, potentially. But then I'm hoping that our work here with TWiEVO will allow me to score the TWiEFecta the hard way, so to speak.
It could be. It's a lot more work. Yeah, you have to be on many podcasts, but that's a good way to do it. Oh. The TWiEFecta. So TWiEVO, I like TWiEVO. There used to be something called Evodevo, right?
Yeah. Exactly.
Does it still exist, or is it kind of subsumed? It does.
No, it does. So, but it's a really interesting point that you bring up. So Evodevo, I would say, which is the blending of evolutionary biology with developmental biology, sort of rose to prominence in the last 20 years and has, some folks would say has plateaued out a bit. But again, that field sort of being invigorated the same way that cell biology is now doing the same thing with evolution. And so, this idea of these collisions of scientific worlds, so to speak, I think opens up all of these interesting new possibilities.
So, what is evolution, Nels?
Well, if we turn to...
Is that too hard a question?
No, not at all. So if we turn to the textbooks and just look for a simple, quick definition, you can find things like changes in heritable traits over successive generations among living organisms. And that idea actually traces all the way back to Darwin. He was a little more poetic when he called evolution descent with modification.
I love it.
I do too. I agree. And so, even in these simple kind of one-sentence definitions, I think you already get this great idea about the fundamental basis of evolution, this idea that, as we proceed through replication or reproduction, the successive generations, that there are these changes that are occurring, that are some of them very fundamental to innovation and progress, others that are just sort of hitchhiking along the way. And that by sort of listening in to that record or trying to find some meaning or explanations in how the process unfolds, that it sort of holds the keys to understanding how we as species tick, as our new motto goes. Mm-hmm. And so this then, what is evolution? I think it ranges all the way back to questions about origins of life. Where did life emerge from? When did it emerge from? But then, up until today and going forward, the ongoing changes that continue to happen and emerge all of the time.
So since Darwin's day, people have been studying evolution, and I assume that the way you study it has also evolved over the years as well, right?
Yeah, that's a really good point. So, classically, I think evolution was mostly, because it had to be, was mostly made up of pure observations of nature. And this included living things, but also fossils, right? And so I think that's sort of a comfortable association with evolution, is sort of exploring the fossil record to try to look back in time, basically. And then just as you're saying, this idea of evolution proceeding in terms of how it's studying. So as we move forward from Darwin's time and even to around the turn of the century, there's a really big push in genetics, and that is manipulating organisms. This is still, remember, before anyone knew about what the material that makes up genes. So DNA had the structure of DNA, and its implications had still yet to be discovered. But people were already manipulating organisms. Fruit flies, in particular, stand out as a model system to cause modifications with descent, so to speak. And then there was also a very big push in mathematics. So looking at population structures, population genetics, and trying to model or predict using equations how that might proceed. And so we end up with then these sort of multiple fields running in parallel, genetics and population genetics, one being very much a laboratory science, one being more quantitative science. And then what we're seeing now, and I think, again, sort of the reason I think for embarking on something like TWiEVO is sort of an increasing collision with people taking experimental approaches to think about evolutionary biology and how things change.
So that's a good point. So basically the Darwin way is to go into nature and look at it, right? Mm-hmm. And you can't really manipulate it very much. And so there are limitations about what you can conclude. But if you then go in the lab and you do fruit flies and nowadays viruses, you can look at it and manipulate things, right?
Yeah, exactly. And so, you and I share a strong devotion to the viruses as a way to study evolution. And viruses are great. So they've been around, or are inferred to have been around since the very beginning, the emergence of life. Certainly since the emergence of eukaryotes, the cells that are single cells that are the origin of what we became in our lineage as mammals, primates, and humans. But viruses, going back to these simple genetic elements, can cause really drastic evolution. So these life and death scenarios where infections might be sweeping through populations and causing very rapid evolution that you can see unfold in some cases right before your eyes, either in the wild in terms of pandemics or in the laboratory where you can control and manipulate things.
Is there any one thing you can point to that you would say revolutionized the field so that now we have people like you in it and Harmeet and Sarah Sawyer?
Yeah, that's a great question. So I think, again, as people were, and we'll actually get into some of these ideas in the paper, which is a review that we'll talk about in a few minutes. As these fields were sort of emerging, and especially the revolution of molecular biology that began in, arguably the early 50s with Watson and Crick discovering the structure of DNA. These fields that have, the training is very different. So as I mentioned briefly at the top, I grew up scientifically as a cell biologist, and so I was really trained in taking an experimental or an empirical approach to studying systems, which then kind of progressed very naturally into microbiology and to virology. Whereas traditionally, the evolutionary approaches have been much more based on inference. And so that collision between experiments and evolutionary inference has really, I think, pushed things forward in new, exciting ways.
So before we get into this paper, in fact, the papers are going to be a big part of what we do on TWiEVO, right?
Yeah.
That should be interesting. And I'm looking forward to learning a lot. And
you and me both.
So I got an email today we get lots of emails on my other podcast, and hopefully we'll get some here too.
Yeah.
I'll tell you later about how you can send those in. I got an email from someone who said, in a recent blog or something, you mentioned something happening 100 million years ago. Mm. And he said, I cannot handle that. I have other views of things, and I don't believe you can say that. He was referring to a virus infecting a wasp, which we think happened 100 million years ago.
Yeah.
The first time these wasp viruses went into the genome. And he said, I really like this field. I'd like to take your lectures, but I have a feeling I'm going to have a fundamental problem with them.
Yeah.
You know what he's talking about, of course.
I do, yeah.
And so there's this evolution is a tinder flame that, sets people in motion because there's some people who can't deal with this idea that things evolve and there's natural selection and so forth. So, we can't answer it, Nels, but I know we're not going to have a lot of people listening because of that. We'll have people listening who are interested in how the field progresses and so forth. But I just want to bring that up. There are a lot of people who, I think you're welcome to your beliefs, and you have to understand that there's great evidence that science happens in certain ways, and we don't want to exclude you. So how do we deal with that?
Yeah, great. I'm glad you brought that up, Vincent. It's a really a great and I think honest response. And to be completely honest myself, I mean, how do we wrap our heads around what 100 million years even means? When we think about our entire lives that might stretch from 80 to 100 years, and how to compartmentalize that, start thinking about the orders of magnitude to 100 million years, and you can, I think, very quickly understand why people would certainly grapple with someone coming to them with an idea that's so wild and incomprehensible in some ways. But that's where, evolution, I think, is really, again, in some cases based on inference that can then be strengthened by empirical or experimental approaches, or sort of gathering a whole range of different kinds of evidence to make a conjecture like that virus that showed up in a wasp genome. Mm-hmm. And so we'll talk a bunch more moving forward about how we estimate times and date things based, again, on fossil records and divergence in mutation rates or so-called molecular clocks in the DNA that starts to get us to those what can seem like on the face really outlandish numbers. But then as we start to look at all of the evidence, both fossil record, DNA, and then increasingly include some experiments, then I think it starts to actually really become, if you step back, it starts to become much more possible and does not sound so outlandish. But at the same time, I think another point that's sort of in that question that you're raising, which is also important, is that because evolution sort of tackles these ideas about the origins of things, where things came from, it can really collide in some ways with people's thinking or philosophies about their religious beliefs, obviously. And so that's kind of an ongoing and interesting controversy, is the collision of these things, science and religion. And I don't think we'll get into it a whole lot today as we're sort of just trying to introduce what we're doing, but I'm certain we will grapple with that a bit as we continue with the podcast.
And please, if you're of that ilk and you want to listen, do listen. We're not going to condemn you or criticize you for your views, but what we'll do is we'll try to present the science in an unbiased way. And these are the experiments, these are the results that we get. You make your own decisions, but this is what we think, right?
That's absolutely right. And I think we're really here to reach out and try to promote healthy conversations. And so I certainly, and I'm sure you feel the same, Vincent, anything, let's put it on the table and
Absolutely.
just have an open discussion.
No, if you have a question and you want to challenge us, please do. Just don't be nasty. There's no reason. All right? We're just scientists who want to find things out. We're really curious. I mean, this is the key. I've always telling people when people say, what's the one thing that's the most important? And I say to have, and it's curiosity.
Yeah, absolutely.
You need to want to know how things work around you, right?
Yep. And once you start kind of following your nose or chasing that curiosity, things can get really interesting.
That's right. All right, so you picked this paper. Why don't you start telling us about it, Nels?
Yeah, I'd love to. So the paper we're going to tackle today it's a review that came out about 8 years ago. It's titled Mechanistic Approaches to the Study of Evolution, the Functional Synthesis. The paper is, I think, really an interesting way of sort of setting up some of these scientific collisions that we've been alluding to already. It was written by two really interesting scientists, evolutionary biologists, biochemists, I would say, and just great communicators. This is Tony Dean, who's a professor at the University of Minnesota, and Joe Thornton, who was at the University of Oregon when he wrote the paper and has now moved his laboratory to the University of Chicago. The paper showed up in a journal called Nature Reviews Genetics, and we will post it to the website for those who want to dig in and really take a look at it. So, and I thought, Vincent, it might be fun just to take a moment to consider just some of the personal connections I've made over the years with these two authors. I think it just illustrates how our business of science is still, it's a growing in community, but it's also still on a scale that it's a very personal community. You can interact with folks and learn things from each other, which is really fun. So about the time this paper came out, I was a postdoc, just starting my postdoc, as I mentioned, in Seattle. And the paper really resonated with me, sort of putting forth these ideas of combining evolutionary approaches with experimental approaches. And so I noticed that this fellow Tony Dean was coming to visit Seattle, and I just shot him an email. And before I knew it, he and I were sitting at a bar in the University District by the University of Washington discussing some of my work and his ideas. And it was just really inspirational that someone who wrote such an interesting review about the field, in my mind, would just sit down with someone who was just kind of getting their start. Tony turns out to be a really fun and outgoing guy. He's well known for his eclectic T-shirt collection, which includes a lot of, let's just say, politically incorrect, in some cases, but humorous T-shirts, sort of a colorful character.
Nels, I'm not surprised that this started in a bar, because that's where TWiEVO started, right?
Oh, good point, yeah.
You and I were in a bar on the last night of ASV, right? And
that's right.
I think it was called Toboggan.
That's correct.
In
London, Ontario.
Right. Mm-hmm. And I said, do you want to start a podcast on evolution? And what did you say? I
think I said yes after taking a stiff drink and stealing my courage.
There are just some people that would be good at podcasting. And what I mean by podcasting is simply talking about what's interesting to you.
Yeah,
right?
Well, yeah, we'll find out. I mean, I have to say, Vincent, when you made the offer or brought up the idea, I was absolutely thrilled. Given your stature among scientific podcasting, which actually you've been asking a lot of questions. I'd love to ask a question here too, which is what got you into this podcasting in the first place?
So as you know, I've been here 33 years, been running my lab studying viruses, right? I've had a traditional career. About, 15 years ago, I got involved in writing a textbook, Principles of Virology, which is now in the 4th edition. And the way this was written made me learn a lot about other viruses that I didn't work on. So after having written the first edition, I had all this knowledge and I wanted to share it. And at that time, blogging had just started. So I started blogging. This is now 11 years ago. And it was very surprising to me how many people you could reach by doing this thing on the internet, right? So I kept doing that and doing it. Meanwhile, I have a very long commute. I drive 38 miles each way, home and back. And I got tired of listening to the radio. I got tired of NPR. And then I discovered podcasts that you could listen to what you wanted to. And I said, this is very cool. I was so excited. And then at one point on a podcast I was listening to, the fellow said, and this guy's name was Leo Laporte. He runs a podcast network called This Week in Tech, TWiT. He said, if you are passionate about something, you can podcast. And I said, yes, I am completely passionate about viruses. Cool. And I want to podcast about it because I can do it. I can sit down and talk. So that started This Week in Virology, which I named after his podcast network. And in fact, a year or so in, he actually reviewed TWiEVO on one of his shows.
Oh, wow.
It was very, very exciting. And so we've grown and it did well. People listened. So I started a few others. And so we have This Week in Virology. We have This Week in Parasitism. We have This Week in Microbiology. We now have, and we also have Urban Agriculture, which explores indoor urban farming.
Yeah. And
now we have TWiEVO. And I do have ideas for other podcasts as well. I have a couple in my head. At some point I'm going to close my lab in the future, maybe in 5 years or so, and I'm just going to podcast. I tell you, I love talking about science.
Yeah.
I love sitting down and having these conversations. I love inspiring people.
Yeah.
People seem to be really inspired. We get lots of emails. I'm hoping we can interact with our audience as well. I view this as a cool thing because I've sort of made a change in my career. I've had a second career, which I think is always good to do something different, especially after you've done something for a long time.
Yeah.
And I can influence people. I can make them be interested in science, change their careers. The people have been wonderful in that sense. So, and I have all great co-hosts and so forth. So that's why I do this, and I will do this till I drop for sure. And hopefully I can fix it in a way that it continues after I'm gone and other people can take over.
Very cool.
But I love it. I absolutely love podcasting.
It shows.
That's why I do it.
And speaking of sort of multiple careers or making pivots in a career, that actually takes us really nicely to the second author of the paper we're considering today. So this is Joe Thornton, who I mentioned is now at the University of Chicago. So the interesting thing about Joe is that he was an English major. He didn't take his first course in biology until he was 30 years old. And before that, he worked for Greenpeace. Very interested in pollution and fighting or countering some of the toxic substances that end up getting released into the environment. Then he made his big pivot about age 30 and ended up getting a PhD at the American Museum of Natural History, actually in your neighborhood there, associated with Columbia University, where he also did his postdoc with a fellow named Rob DeSalle, who's in the ecology evolution area. And so there's a great example, I think, of, and actually this is a recurring theme in evolution, is that there are lots of people who sort of make a late pivot to just get sort of captured by evolution and end up changing their careers in dramatic ways to do that. So we mentioned briefly my postdoc advisor, Harmeet Malik. He was actually on the road to becoming an electrical engineer before he made that pivot into genetics and evolutionary biology. And so there's all these sort of interesting, kind of paths to this area of it, that I think is really fascinating. Also, speaking of fascinating things, so Joe Thornton, I was just peeking at his website, and he notes that he also plays in an adult hockey league where he's claims that he's the leader in terms of accruing penalty minutes. So
stay away from Joe.
Stay away from Joe. Sounds like an outlet here.
Do you play hockey, Nels?
I don't. So, as I said, I grew up in Minnesota, and so avoiding hockey, you kind of have to go out of your way to do it.
You're right.
I played basketball.
Yeah, you're a tall guy. That's right.
Yeah. So that's sort of one of the things I like to do.
Cool.
Yeah. So then why don't we jump in here on this paper? So the title, as I said, is Mechanistic Approaches to the Study of Evolution, or the so-called functional synthesis. And the authors start to explore this idea of the divide between that sort of historical divide that we just hinted at between evolution and molecular biology. Had you seen this paper before, Vincent?
I had not seen it. I really enjoyed it because I didn't really know that the two fields had split so severely, this schism that between evolutionary and molecular biology, because I was trained as a molecular biologist, right?
Yeah, me too.
And, we had DNA, and then we had recombinant DNA, and so I applied that to viruses, but I just studied, one or two viruses, and they were laboratory specimens. I knew there was a field called evolution, and I associated it with dusty fossils and, Chuck Darwin and his finches. But then, a couple of years ago, maybe 5 years ago, guys like Harmeet and you and Sarah Sawyer and many others, all of a sudden are doing cool molecular biology with evolution. So yeah. I just didn't realize that this was a split to begin with. So this paper puts it in a nice view. I really like it. And here's one of the cool things that I liked. A great strength of molecular biology is the high standard of evidence-based inference. Molecular biologists set aside much of the complexity of nature. That's exactly what I did with poliovirus.
Yeah.
I took one virus and mutated it and studied it in cells and ignored all the other viruses like it that are out there. But now a guy like you might say, Vincent, why don't you get 100 isolates and see what's different and study that, right?
Exactly. And so that's where the authors, I think, really nicely make that contrast between exactly what you're describing and how I grew up in science, which are these reductionist approaches where you try to isolate one variable and keep everything the same, versus evolutionary biology, which embraces the complexity in nature. So making those observations in the wild of, Darwin's finches with all the difference in beak morphology, and instead of trying to simplify those things to actually learn something about that complexity. Both approaches, I think, are amazingly powerful and interesting, but they both do different things well. And so that's what the authors, I think, are doing here, is trying to highlight how the merging of the two, the collision of them, can start to lead to interesting stuff.
I think it's interesting. They say about evolutionary biology before the merging, claims that are based solely on associations remain standard in the field. Mm-hmm. So in fact, the finch story of Charles Darwin is really an association story. He didn't do any experimental work to prove it, right?
Yeah, that's right. And, at the outset, too, the idea of association, I think, one thing to avoid, or at least I try to avoid in my thinking about this, is not to think of one approach as being stronger or better than the other. And in fact, even today, a lot of evolutionary biology continues to be driven in really interesting and new ways through increasingly sophisticated statistical associations. Mm-hmm. A lot of human genetics certainly falls into that category, with the revolution of genome sequencing and being able to make not only tens of comparisons or hundreds of comparisons, but millions of comparisons, and to try to infer something interesting from that data. But at the end of the day, there is something to be said about what a strong association looks like versus what a falsifiable hypothesis is in terms of the evidence.
Nels, can you give us an example of the prefusion evolutionary biology? Give us an example of an experiment that was based on associations.
Yeah, I'd love to. So, and let's go, Vincent, all the way back to around the time of Darwin. And one of my favorite classical evolutionary sort of approaches. And these are again, just observations, but really sophisticated observations of nature. And the experimental or the evolutionary biologist I'm thinking of is a fellow named Henry Walter Bates. He was in the Brazilian rainforests just looking at butterflies. And what he started to notice was these really interesting patterns, which are, I think to a layperson would be, it's staggering to think how he was able to have such a discerning eye. But basically, he was looking at butterflies that were known to be inedible to predators. So these are birds and hawks and so forth that will avoid butterflies because they're poisonous. And eating them, obviously, is not a great experience for the birds. But there are plenty of edible butterfly species that birds avoid as well. And what Bates figured out was that part of this process was that the edible species were mimicking the poisonous ones in their physical appearance. And so even though these butterflies looked identical, basically, to birds who thought they were poisonous, the butterflies couldn't mate with each other. And so Bates inferred that this really cool evolutionary observation that these edible species could gain an advantage through descent with modification if that modification meant that they looked or resembled these poisonous species. And then as you move through the generations, they might come to resemble the poisonous species even more and more. And so there I think we have just an absolutely beautiful set of sophisticated observations associating the mimicry of unrelated butterflies as a evolutionary innovation.
How would you take this to the next step with molecular biology?
Yeah, so then, I think another important example here that we should really get on the table and sort of put into evidence is the idea that's sort of fundamental to evolutionary comparisons, which is what are the relationships or comparative relationships between species. And so the classic way that I think a lot of people are familiar with in terms of visualizing this is the so-called evolutionary tree or tree of life or phylogenetic tree. And so here is a way of visually representing the relatedness between species by drawing branches on a tree where branches closer together symbolize things or species that are more closely related to things that are on branches more farther apart. And in fact, that kind of visualization or way of conceptualizing goes all the way back to Darwin. So I'm sure, do you remember seeing that kind of rough tree with the scribble next to it that says, "I think"?
I do, and I saw it in that bar in London, Ontario as well.
That's right.
Do you remember that?
I do. One of our colleagues had a tattoo.
In fact, maybe that'll be our episode art.
There we go.
I have a picture of that young lady's tattoo on her scapula.
That's right. We should put it up. It's another formative moment in TWiEVO, up in bar in Canada.
Yep. So those trees in the old days were based on similarities and differences, physical ones that you could see in organisms, right?
Yeah, correct. And so these were things like comparisons of fossils to really go back to try to think about the history of these trees. At the same time, people were starting to compare traits of extant or living organisms for differences in morphological characteristics. So, some classic examples are the idea that you have wings in birds and bats, and yet if you really look closely at them, you would not put bats and birds together in close position on the trees because they kind of independently came to those features. And so then, so people continue to do that and build or try to find characters or ways of comparing. And the problem is, the bird and bat example is a good one. Here you have morphological features that are very similar, but can kind of fool you into thinking that there's more of a relationship if you don't look really closely. And so here's where I think, the sort of revolution of molecular biology started to come in very important ways for evolutionary thinking, and that was the advent of molecular characters. So as people first used proteins as markers, so these so-called allozymes, where you could start to segregate features based on how proteins would migrate through gels put under an electric current. But then the real breakthrough came again with DNA, the discovery of its structure and the insights into the fact that this was the genetic code. This is the basis of that descent with modification. What was being modified by and large, is DNA. The base pairs changing their chemical structure, and the base pairs mutating or substitutions accruing. And so now you have this language, once that was deciphered, of DNA, the A, T, C's, and G's, that code for these triplet codons amino acids. And so you have this alphabet, literally, that you could use to build these trees by comparing the letters and how they were assembled in related species. And this really was a breakthrough in terms of, I think, the first sort of collision between evolution and experimental biology or molecular biology was there.
Roughly when is this happening? In the 70s, 80s?
Exactly.
When you started to be able to sequence DNA, right?
Exactly right. Yeah. And so, but, if we step back and think about it, we're still looking at inference without experiments. We're just making stronger inferences because we have sort of a higher resolution view of the relatedness between species. So if we look at some common genes in birds and bats, what we'll see is that they're actually they're much more different than we would have predicted if we would have just used the wings, that sort of gross morphological feature as a character. And so that's when we really started to get a more fine-grained look at the relationship between all living organisms. But this is still an inference. So these are when people put together a tree of life, or a phylogenetic tree, as we commonly call them. It's a hypothesis. It's saying here we're observing all of these changes, and we're using that to form these relationships. But we can't actually physically go back in time in a time machine, and so there are possibilities that some of those changes that we see don't actually reflect the real evolution. And so
Nels, so let's say you have a tree that's based on morphology, right? Pre-DNA sequencing.
Yeah.
I presume some of those, let's say plants, some of the old ones are not around anymore. You're depending on pictures or some sort of thing. And now you want to verify it with DNA sequencing. But you don't have any of the old plants left. What do you do?
Yeah, great question. So and this is one of the interesting things that emerged from phylogenetic analysis, or so-called building these phylogenetic trees, is that as you make multiple comparisons, you can actually infer what you think the sequences were in those fossil records, what the ancestral plants that aren't around anymore. And basically what you do is you start to look at shared changes between things that are more closely related, and then drawing a node between those and something that's more unrelated. You can start to predict, based on those at the very simplest level, using those three sequences, what the ancestral sequence looked like. And then as you get more sophisticated and make more comparisons, and as folks have applied more elaborate statistical methodologies, now we can actually, with some confidence, depending on how good your data set is, actually infer what the actual ancient DNA sequences looked like. And that comes up as one of the themes in the review that we're looking at.
And of course, if it's a protein coding sequence, a single protein, you could actually make it and see the properties of the protein, right?
Yeah, which is really, so again, this collision of evolution with experimental biology. This is one of the really exciting breakthroughs, is basically this ability to not only predict what something looked like in the past, but then using techniques of genetic engineering to build it and to test it, to bring it back to life, so to speak, and see how it behaves in the laboratory.
So Nels, if you compare phylogenetic trees made the old way, compare them to those made by DNA sequence analysis, do you often get congruence, or are there differences, or do you rearrange the trees sometimes?
Yeah, all kinds of differences. And in fact, there are this is an entire field. So phylogenetics is an entire field. It's vibrant and involves a lot of quantitative approaches. There are many ways, even using DNA or amino acid sequences, there are many different ways to infer the relationships between species. And so even there are many cases where so you're using the exact same data set, but the way you analyze the data set will lead to differences in how you infer, or the hypothesis that you generate for what the relationship between things look like. And so it depends very much on what you're comparing. So are you picking just a single gene sequence? Are you picking many gene sequences? And are you comparing entire genomes? There are sort of trade-offs in strengths and weaknesses to all these approaches. And what you end up with in what is an actively researched field is this scenario where you can draw many different trees to explain the same data set. And so what folks are trying to do is to get more confidence in by, again, comparing approaches or developing new approaches, that the hypothesis or relationship you're proposing is actually reflects what the true evolutionary history was.
Yeah, maybe we'll do a paper on that in the future.
Yeah, that would be fun. That's definitely a open and ongoing area.
All right, I'm asking enough questions. Get into this paper.
Yeah, we are. So,
do you think we're going to try and limit these TWiEVOs to an hour?
I think that's a good idea.
All right, yeah, we got 15 minutes.
Yeah, I know. On the pace we're going, we'll just be podcasting for the rest of our lifespan. So we should move ahead here. So the cool thing about this review, and why don't we skip ahead a little bit, is that so in addition to, and I'd really recommend that people read even if you don't have a great background in biology, just reading the kind of historical stuff at the beginning, I think it just frames the field of evolution, contemporary evolution, in some interesting ways. But the other thing that the authors do is that they give us 4 or 5 kind of example cases of how experimental biology and evolutionary biology have merged. And so I thought, why don't we just do 1 or 2, depending on our time, as some examples of what it means to sort of collide these scientific worlds.
Sounds great.
Cool. So the first one is a story about insecticide resistance that comes out of John Oakeshot's lab. This is a lab in Australia. And they were looking at the sheep blowfly. Have you seen these flies before?
I have not.
Yeah, me either. I did a quick Google search. They look a lot like a housefly. And yet the reason blowflies get the attention of biologists...
they do, yeah. Lucilia cuprina.
Yep, that's right. So they look like kind of harmless houseflies, but the reason they get the attention of both biologists and farmers is that they have a devastating effect on sheep. They will lay their larvae in sheep, and this can cause real health problems. And so I think it was about the mid-50s when people started experimenting with releasing insecticides. The one that they mention in the review is one called diazinon. And this insecticide would act to kill blowflies. And so that was, and soon after they released that, I should mention, what they saw was that blowflies, and this is evolution in action, started becoming resistant to that insecticide. And so the authors of the studies that were looking at the blowflies were interested in, well, what happened evolutionarily or at the genetic level that made blowflies resistant to insecticides. And so what they ended up doing in a series of papers that are just summarized in this review is they started grabbing blowflies that were both sensitive to the insecticide or resistant to the insecticide, and they started sequencing genes that they thought would be associated with this. It turns out some earlier work had suggested a specific gene called an esterase that might be the source of this resistance. And what they saw was that if they compared, again, the alphabet, the DNA letters of the gene that encoded that esterase, this enzyme, that there were 5 differences common to either the resistant or the sensitive blowflies. So that gets them, in a sense, to a strong association, right? So if you're a resistant blowfly, you have in this one gene 5 differences in DNA letters that lead to 5 differences in protein letters. And so the next question then becomes, however, what's the experimental evidence, or how do you test whether any of those 5 or all 5 of those changes are what makes a blowfly resistant or sensitive to this insecticide? And so that's where experimental biology or molecular biology came in. They were able, using genetic engineering, to one by one make these substitutions. And what they saw was that they could narrow it down to just a single amino acid change that would explain the resistance or sensitivity of the blowflies to this chemical, the insecticide. And then from there, the authors actually did something that we see a lot in these combinations of molecular biology and evolutionary biology, is they turned toward the structural biology. So there was some really, at the molecular level, the atomic level, there was information about what this enzyme looked like. And so they were able to map onto that structure this single change and see that it was in the active site of this enzyme. So again, they're starting to build a stronger and stronger case by actually experimenting and making further observations in addition to the evolutionary approaches. And here's where I think the story even gets more interesting. So once they had that insight that there was a single change, a single mutation in a single protein that could explain this phenotype or difference between resistance or sensitivity, they actually went back to that complexity, to the evolutionary approach. So there's sort of this, this theme of this back and forth from doing experiments in the lab, but then returning to the wild. And what they ended up doing was sampling more flies. So they got wild flies. They also went to museums that, I don't know, museums actually will keep around blowflies in some of their collections, right? You never know what you're going to be interested in collecting. And so then they could actually look and say, okay, because they had museum specimens, they could actually do something evolutionary, which was to look back in time. Those specimens had been preserved before the time that insecticides were being used in the mid-50s. And so they could ask, wow, when did this amino acid change or mutation show up? And what they found was that the single change they found was not in any of the museum samples they looked at. But when they looked nearby, they saw another amino acid change, which turns out actually gave the blowflies resistance to a related but different chemical from the first insecticide that they were looking at. And so here's where, to me, it really becomes this cool detective story. And this is, again, a theme for a lot of evolutionary studies that mix molecular biology, is that you can then start to make really cool inferences by mixing these approaches. And so as they continued down that path of figuring out what was going on here, what they realized was that the reason they probably did not see that mutation for that first insecticide in the population was because there was a big cost or a trade-off to having that. Mm. And the trade-off is that enzyme, if it has that mutation, is actually not as active as it would be otherwise.
Do we think that other mutation was selected by an insecticide at another time?
To the original one, probably not. So because in that case, so the nearby mutation was just standing as genetic variation, and by chance, it happens they did the tests, the experiments, and what they found was that other mutation doesn't impede the enzyme activity, so it's going to be in the population because it's not going to be culled out or purified away. We'll talk a lot about, down the road, experimental or selective pressures and how we think about that, both as evolutionists and experimental biologists. But so the idea was there was no cost to having that one change, whereas the only way you would come up with the first change that they saw was if you were under this strong selection by the insecticide.
So the diazinon resistance selects for a single amino acid change. Does that single amino acid have a fitness cost in the fly?
Exactly. It does. Yeah. The fitness cost is that enzyme does not operate as well. Mm-hmm. And so the only way you would tolerate that cost was if the rest of the population is basically getting wiped out by diazinon. Right. And the only way to survive is you'll sort of tolerate that cost.
Right.
To make.
So if you take away the diazinon, eventually the mutation will go away.
Exactly. Exactly. That's the prediction.
Right.
And then what you could do is, again, figure out ways to experimentally test that by either setting up in the laboratory sample, blowflies, where some are maintained on the insecticide while others aren't, and then pass them through the generations, or continue to sample in the field in the next, 10, 15, 20 years, depending on if there's exposure to that insecticide or not, see what happens.
So I can imagine in the pre-sequencing days, either DNA or protein, they would describe this resistance. Maybe they could do some genetics on the flies to show how it segregates as a gene, right?
Correct, yeah.
And that would be it. They wouldn't know the basis for it.
Yeah, exactly. That's right. And so, while you can still make these really powerful inferences, you're left sort of on the edge of the cliff, wondering what's really going on here. What is the basis of this? And that's where the merger of these fields, I think, leads to a lot of interesting stuff.
What do you do with organisms that, like humans, that have huge gestation periods and live a long time? And you can't really do this, can you?
Yeah, you're right. And so, as a consequence of that, what we've seen is really an explosion in the resources that we put into making better and better associations. And so that sort of, underlines the genetics or genomics revolution, where what we are doing is as fast as we can sequencing more and more human genomes from more and more distinct populations to then try to make inferences.
And of course, you can take an individual gene from a human and make the protein and look at its properties and see the effects of mutations that you've seen in the population. So those sort of things you can do, right?
Exactly. Yep. And also this idea of model organisms. So, not only to put it into a dish, say with, human cell lines or something like that, but also rely on the fact that, when we build these trees, we can actually put ourselves on the tree and see close relationships between other primates, other mammals. Mouse is an example of one that's used very commonly in genetics research to stand in as a proxy for human biology. And then we can keep marching out the phylogenetic tree to things like fruit flies and worms and zebrafish and yeast and even viruses that are useful tools for thinking about how we get at our own biology.
Right. All right. You want to do one more example?
Yeah, let's do one more quick one. And so this story is the evolutionary basis of coat color changes in mice. And these studies have been spearheaded by a few groups, but the one that they highlight is Hopi Hoekstra, who is a really interesting evolutionary biologist. She is a professor at Harvard in the Department of Zoology and Evolutionary Biology. She's also the curator of mammals at the Museum of Comparative Zoology. And so for many years, going back to her postdoctoral training in Arizona, she became interested in these color changes that you see by just going out into the wild and looking at beach mice. These are mice of the same species that, depending on where they live, whether they're living in a sandy environment or a rocky environment, they look drastically different. The ones living on sand are light-colored. The ones living on rocks are dark-colored. And there's been a lot of ecological work that has really suggested or associated those differences in color with an imperative to avoid predators through camouflage. So you're a light-colored mouse on sand, you're basically invisible to a hawk flying overhead. If you're a dark-colored mouse on rocks, same thing, but on the dark rocks. And so here, is another emerging theme that I think we're going to be seeing over TWiEVO, is the influence that competitors, predators, pathogens, and even viruses have on their hosts. This drives a lot of really interesting evolution. So Hopi in her lab, they became interested in what's the genetic basis then of these differences in coat color, this very conspicuous phenotype or difference between the mice. And so they used, again, sort of a candidate gene approach. They had some clues based on previous work about what might be the genes that have differences that might be the genetic basis of the color differences. And so they focused on a single gene called melanocortin 1 receptor, or MC1R. This is the alphabet soup of genetics. And what they did was simply isolate that gene from light-colored beach mouse and dark-colored beach mouse, and they saw a single amino acid change. All of the light-colored guys had one letter at a certain position in the protein, whereas all of the dark guys had a different letter at a single position in the protein. Everything else was identical. And so just as a little background on the biology here, this melanocortin receptor sits on a cell and waits for a hormone. These are, in this case, it's a small hormone that stimulates pigment production. That's the coloration. And so that's kind of already linking or evidence of these things sort of coming together. The hormone is a peptide, a short protein. In mammals like mouse and human, it's produced in the pituitary gland. That short hormone peptide will bind to this receptor, MC1R, and that causes a reaction where the pigment that's inside of the cell will move towards the edge of the cell. And if you do that on an organismal level or tissue level, the mass action of moving all of these so-called pigment granules will cause the organism to be darker. These things can be put into hair or skin, depending on the animal that you're considering. And so already the biology was starting to connect with the evolutionary observations they were making. Then what they did is they went on to actually express those variants of MC1R, so that's making that single amino acid change, and putting it in cells and tissue culture. So just like we were talking about with humans, you can't really experiment on humans. Same here. It turns out the easiest thing was to just put the different proteins in cells, and they measured different activity. And so there again was sort of crossing that threshold from associations to starting to actually see biological outcomes that they were testing in the laboratory. And so it turns out they continue to chase after the biological basis of this. So some other genetics approaches they've done, they found out that this MC1R protein is just one part of the story. And there's actually many genes and variation, presumably, that's contributing to this change in color. And so one of the reasons I thought it was important to bring this example up is that, again, we go from the evolutionary association, the experiments, but now, again, back to exploring the complexity or the wild in nature again. And one of the fun things about what the Hoekstra lab is up to is they're out actively looking at using their genetic insights from the laboratory to look at more complex scenarios in nature. And so a lot of her lab is now actually somewhere in the middle of Nebraska, setting up these massive enclosures where they monitor wild mice that are living on either light sandy areas or dark rocky areas. There are a lot of natural native hawks out there that are swooping down and eating them. And then what they're doing in real time is collecting these mice and looking at the changes in the genetics by sequencing the genomes of these populations. Mm. So I really consider Hopi one of my science heroes, just for sort of the boldness of extending this functional synthesis, doing all of this field work, taking it into the laboratory, going back out into the field. I think it's really exciting.
I was just looking at her website. She's got a big lab, and now I know why, because I think it's probably a lot of fun to combine field work with this kind of precise molecular biology, right?
Yeah, exactly.
So this brings up so many cool things. First, so the variations in MCR1R and all these other genes that you say are involved as well, they must have some other roles, right? Besides just predation. So the pressure, the selective pressure here is predation. You know, if you're white on a rock, you get eaten. But if you're darker, you have to be if you happen to have been made darker for some other reason, maybe, right?
Yeah, good point. And that we'll be talking about this a lot. So you have these so-called pleiotropic effects, or something that it's very easy for us to look at the mouse on the beach and say, oh wow, all of your biology must be geared towards being a light color. But life is, like in all situations, is much more complex, and we only see what we're looking at. And so in a lot of these cases, and we saw it actually in that first example we talked about, the insecticide. If you made a change, there was this trade-off where now an enzyme that has an activity is not working as well, even if you survive the insecticide. So we're seeing this is one of the things that's really emerged as a theme time and again in the functional synthesis, is that there are all of these mutations that could very much have been influenced by predators, but are influenced by a lot of things, or in a lot of cases aren't influenced by anything at all. It's just random chance that they're there. And so as modern experimental and evolutionary biologists, you have to take a really broad view of what might be going on before you get too sort of enamored with one of these so-called just so stories where everything explains things in sort of a neat packet. So
the William Proxmire's may say, why do we care about studying mouse coat color?
Yeah, that's right. And so
you know who William Proxmire was, right?
Actually, I don't.
So he was a U.S. senator.
Oh, right.
Who used to give out the Golden Fleece Awards.
Oh, yeah.
And he did that for research that he thought was really a waste of money. And some of them sounded pretty bad. He did identify some nonsense, but He also, things like fruit fly research and maybe mouse coat color might fall under his. So tell us why this is important.
Yeah, good point. So now that you bring that up, I do remember this fellow, and and some of the nonsense I would say that's coming from that viewpoint. So it's true. On the face of it, you could say, "Wow, why would we invest so much time and energy chasing mice around in the field to figure out what effect predation has on the genes?" But we have to remember. So we're all this again. I think is the fundamental part of evolutionary theory and evolutionary biology. Is we're all tied together through common ancestry. This is the basis of being able to build these trees. And so, for example, not only do mice have this gene MC1R, that pigment receptor, so do humans. And in fact, if we look at mutations that are circulating in human populations, they can lead, in M1CR in particular, they can lead to human diseases. They can lead to differences in hair color. One famous example with this gene in particular is the origins of red hair in humans. And so even something that at the first seems very disconnected from our daily lives, actually, because of these evolutionary connections, has everything to do with our daily lives, where we came from, where we're going, and how we move forward as a species. So it's true. I think, it's very easy for politicians sometimes to cherry pick these examples that look like, wow, if you weren't really thinking about the big picture, this looks kind of ridiculous. In every case that I know of, if you just dig in a little bit deeper and listen to the story and the work that the scientists are doing, it doesn't take long before it becomes very clear that we're learning important and vital things about not only our history, but about our future.
This paper actually stimulated a lot of questions on my part, and they're all in the show notes. And I think that these will become subjects of future TWiEVOs because a lot of them are important concepts, right?
I agree with you, Vincent. I think we're just kind of breaking the ice here. And perhaps in the interest of time, we should save some of these great questions moving forward. Yeah.
Do you want to have as a tradition on TWiEVO a science pick of the week? I bet you'd love to do that, right? You
read my mind. So borrowing from your TWiV franchise of, or Twix franchise of podcasts, I thought it would be great if we could do picks of the week.
Sure. For
mine this week, I picked this fellow Neil Shubin's recent series that aired on PBS called Your Inner Fish. And the reason I'm picking it is because it was just this week received an Emmy Award for being a really outstanding documentary, bringing science to life, really, and in very cool ways, connecting the fossil record, the ancient paleontology, with modern biology today, and thinking about what the implications for what it means to be human and how we've come to look how we look. So I think this actually also gets at that last question of yours about, what's the use of thinking about evolutionary ideas. So Neil Shubin himself is trained as a fish paleontologist, and he's famous for these really incredible expeditions where they've found really interesting fossils that help piece together major transitions in the forms and structures of fish and reptiles and primates moving forward. And he's now a human anatomist at the University of Chicago. He teaches medical students. And he really does an incredible job in this 3-show series of connecting these seemingly unrelated topics of evolution with how we live our lives today. So that's my science pick of the week. And we'll put up the link to the PBS website.
Sounds great. I have to check it out.
Yeah. How about you, Vincent? Do you have a...
I do. So I just finished reading a book that I want to... have as my pick. It's called Prelude to Foundation by Isaac Asimov. So many of you are probably very familiar with the Foundation series that Asimov wrote, and I've known about these since I was a kid. You must know Isaac Asimov, right, Nels?
A little bit, I do. Yeah.
Have you read any Asimov at all?
Just a bit. I'm not a big enthusiast, but now I'm glad you're pointing me in this direction.
So my son for Christmas or my birthday last year, one of my sons gave me Prelude to Foundation, which is supposedly the first in the series. and I just finished it last night. This was written a long time ago, science fiction, but it is so prescient. It could have been written today in many ways.
Very cool.
So the story is wonderful. It's about a mathematician who feels that he's found a way to predict the future. And he's living in far in the future in a very different world. And his adventures, what happens to him, are just riveting. So I am highly remiss for not having read more Asimov before. But
me too.
This got me started, and I'm going to go on and read the rest of the Foundation series. I have a few of them at home. So I know a lot of listeners who will eventually listen to this probably know this book, and they're thinking, Racaniello, that was out long ago. But I just, you discover things. That's the way it works.
Yeah, thank you for the tip.
Check it out.
I'll check it out myself. I appreciate it.
And we invite our listeners to submit listener picks as well as emails. And I'll tell you right now how you can do that. So over on TWiEVO, which is where we mainly do our picks of the week on the other Twix podcasts, we have a pick of the week, and our listeners send in picks as well. So please do that. Sciencey things, and that's a stretch. You can go pretty far from science. Send them. Here's the email: TWiEVO at microbe.tv. So TWI, This Week in Evolution, right? TWiEVO at microbe.tv. Microbe.tv is a new website I'm building where I'll house all of these podcasts in one place. They've been scattered all over, and I thought it was about time to unify them. And hopefully by the time you hear this, it'll be live. So you can send your listener picks, your questions, and your comments to TWiEVO at microbe.tv. Eventually, TWiEVO will be on iTunes, of course, and you'll be able to subscribe to it using any of your iPhone or Android podcatcher apps, so you can listen to it everywhere. So Nels Elde, my co-host on This Week in Evolution, you can find him at cellvolution.org. You can also find him on Twitter. ELEARLY bird. Early bird, except there's an extra EL at the beginning, right?
That's right, Early bird. And actually, I should just say, to make it even more confusing, so my last name is pronounced LD.
LD.
Yeah, ELDE.
I've been saying it wrong the whole podcast.
Well, it's the first podcast, so I still had plenty
LD,
of time.
Nels LD, with a real heavy accent on the LD.
That's the American pronunciation.
What's the right pronunciation?
Depends. So like evolution, what matters is the environment. So if you're in the U.S., it's LD. If you're in Europe, it's Elde.
What do you prefer?
I go by LD.
Nels LD.
Yeah.
Should we? Let's re-record the whole podcast, and I'll do it right.
Yeah, right.
Nels LD. Cellvolution.org. That's your lab site, right?
It is.
Earlybird on Twitter. All right, check him out. Thank you, Nels, for joining me today and doing this.
Vincent, this has been a blast, and I have to say I'm looking forward to the adventures ahead.
Yeah, I am too. I've learned so much already. There are a billion things we can talk about. It's a lot of fun, and we're going to have a good time, and I think our listeners are going to enjoy it as well.
Can't wait.
I'm Vincent Racaniello. You can find me at my blog, virology.ws, and from there you can find all the podcasts, or at microbe.tv. You can also find me on Twitter. I am PROFVRR. You've been listening to This Week in Evolution, which is all about the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.